Hi and welcome back to Dental PYQs. Today we're going to discuss the October 2025 biochemistry paper. So let's begin. So let's see the first question of the biochemistry 2025 paper. Describe the sources, RDA and biochemical functions of calcium and add a note on regulation of serum calcium. The sources will carry 1 mark, RDA will carry 2 and the biochemical functions will carry 2 marks. Now the biochemical functions is very important because it has been asked repeatedly in most of the papers. For example, December 18, March 22, December 22. So it's a frequently asked question. And add a note on regulation of serum calcium. This was again asked in August 23 paper. So don't miss out on the calcium because it's very important. We'll write a brief intro. It's the most abundant mineral in the body. The total content of calcium in the body is about 1.5 say 1 kg. 99% of which is present in the bones and teeth, and the rest 1% is present in the extracellular fluids, that is mainly blood. The normal calcium level is about 9 to 11 mg per 100 ml of blood. This is very important. Coming to the sources of calcium, we have the best source and the good source. Best is again milk and milk products. Good sources, we have beans, leafy vegetables, fish, cabbage and egg yolk. Coming to RDA, an adult would require about 0.8 grams of calcium per day. Children will require about 0.8, say 1.2 grams per day. And pregnancy and lactating women will require about 1.5 grams per day. Coming to the biochemical functions of calcium, they are required in the development of bones and teeth. Calcium will be required along with phosphate so that it forms the hydroxyapatite, which is the main content of the teeth. It provides strength to the bones and it also acts as a reservoir for calcium. As we know, the osteoblasts are the bone forming cells and osteoclasts are the bone resorbing cells. Calcium is also required during muscle contraction because it binds to the troponin C. It's required during blood coagulation because it acts as a factor 4 and also for enzyme activation. It's responsible during nerve transmission because it's required for the release of neurotransmitters at the synapse. It's also important in maintaining of the membrane integrity and permeability. Like we have the intracellular and extracellular. Extracellularly, it will bind with the phospholipids. Intracellularly, it's responsible for opening of the potassium channels. It's responsible for activation of enzymes like the pancreatic lipase, ATPase and succinate dehydrogenase. We have cammodulin mediated actions. Now the calcium will bind to this cammodulin and activate enzymes like agenylate cyclase and calcium dependent protein kinases. It acts as an intracellular messenger. Okay. It's also responsible for release of hormones like the insulin, PTH and calcitonin. Calcitonin is nothing but active form of vitamin D. We have the secretory processes which regulates the endocytosis, exocytosis, cell mobility via the microfilaments and microtubules. Contact inhibition is involved in the cell adhesion and cell-to-cell communication. It also has action on the heart while it acts on the myocardium and it prolongs the systole, also responsible for arrhythmias. So you have 12. You can remember how many ever you want, but a minimum of 5 is a must. Then how is calcium dispersed in the blood? We have distribution of calcium. We have it in the ionized form. We have it in the complexed with anions and we have bound to the protein. About 40 say 50% is present in the ionized form or the free form of calcium. Complexed with anions is only about 10 say 15%. Okay, it can be complexed with phosphorus. Then it can be bound with proteins, that is about 40 to 45% like albumin. Coming to the regulation of serum calcium, 3 hormones are responsible. That's the calcitrol, the parathormone and the calcitonin, which will regulate the plasma calcium levels or maintain the homeostasis of calcium. Now, vitamin D, an active form of calcitrol, as we know, it is regulated when there is hypercalcemia. So, this will increase the absorption of calcium from the intestine. It increases reabsorption of calcium from the kidneys. It increases the osteoblastic या bone forming activity in children, and it sometimes also causes bone resorption whenever there is hypocalcemia. Coming to parathyroid hormone या PTH, it's also regulated as when there is hypercalcemia. It increases the synthesis of calcitrol and it increases the calcium absorption from the intestine. There is increased reabsorption of calcium from the renal tubules and increased bone resorption या demineralization whenever there is deficiency of calcium. Calcitonin is secreted from the parafollicular cells of the thyroid gland or the C cells. It has an hypocalcemic effect on the blood. It decreases the reabsorption of calcium from the kidney and it decreases the resorption. Basically, it increases the bone forming and decreases the bone resorption activity. One diagram which shows the interrelation of the intestine, the bone and the kidney. You can write clinical aspects. They always fetch you extra marks in case you have missed out anything, or generally also. The clinical aspects of calcium metabolism you can always write as hypercalcemia and hypocalcemia. Hypercalcemia is when the serum calcium levels are greater than 10.5 milligrams per deciliter. The common causes can be hyperparathyroidism, malignancies like bone metastasis, myeloma. Vitamin D, when there is excess, prolonged immobilization or static conditions. We have thiazide, diuretics and sarcoidosis. The neuromuscular symptoms will be weakness of muscle, there's decreased reflexes and fatigue. The CNS symptoms will include confusion, lethargy, depression, coma. The clinical case clue as to how you will identify a person with hyperthyroidism clinically would be bones, stones, groans and psychiatric overtones. That means whatever is excess. Management would be acute, that's IV normal saline, loop diuretics like furosemide, calcitonin and bisphosphonates. Long term would be treat the underlying cause, stop calcium vitamin D intake and parathyroidectomy in severe cases. Hypercalcemia is decreased of the serum calcium level below 8.5 mg per dl. This is seen in hypothyroidism, vitamin D deficiency, chronic kidney diseases, malabsorption of calcium, acute pancreatitis and massive blood transfusion. The neuromuscular symptoms would include the muscle cramps, tetany, paresthesia and increased reflexes. CNS symptoms would include irritability, anxiety and seizures. The clinical case clue for hypocalcemia would be Schwartz sign and Trocher's sign. Schwartz is nothing but twitching of the facial muscles. Sign is nothing but spasm of the carpals, fingers and wrist backwards, called as carpopedal spasm. The management would be IV calcium gluconate. Long term will be oral calcium supplements, vitamin D supplements and treat the underlying cause. So this completes your 10 marker on calcium. Let's move on to the next question, which is digestion and absorption of lipids. This is asked for 5 marks. This also is a repeat question, which was asked in March 2022 and November 2023. So it's already been asked 2-3 times, so it's important, don't ignore. Coming to the definition of digestion, which says it's a process involving the hydrolysis of large and complex organic molecules of foodstuff into smaller and preferably water-soluble molecules which can be easily absorbed by the gastrointestinal tract for utilization by the organism, basically for energy generation. So more than 90% of the dietary lipids are fats and oil, that's triacylglycerols, and the rest is made up of cholesterol and phospholipids. Coming to the digestion of lipids, this includes digestion of triacylglycerols plus phospholipids plus cholesterol esters. This will be covering separately and these two will be covered together. Lipid digestion possesses a problem because lipids are insoluble in water, but the lipid digesting enzymes are always present in the water medium. So this problem has to be overcome by a process that is called as emulsification of fat, which is usually done by the bile salts in the intestine. Very little digestion takes place in the mouth because the lipids are not emulsified. So the lingual lipases that are present, they cannot act on them. Now this lingual lipase are secreted by the von Ebner's glands from the tongue. Digestion of triacylglycerols. The digestion will occur in the stomach and intestine. Firstly, in the stomach, the lingual lipases from the mouth, which were inactive now, they come to the stomach and they are still active at an optimum pH of 2.5, say 5. Okay, it acts on the short chain triglycerides, that's also called as the SCT, which is usually present in the milk, butter, and ghee. This action is more significantly seen in the newborn infants. The digestion of lipids also occurs even during the absence of the pancreatic lipases. Coming to the gastric lipase, it's an acid-stable enzyme with an optimum pH of about 5.4. It's secreted by the chief cells. It's stimulated by gastrin. Bases initiate the lipid digestion by hydrolyzing the triacylglycerols to mainly 1,2-diacylglycerol plus free fatty acid. Now in the intestine, we have a very important procedure that occurs, that is emulsification, which is a prerequisite for any digestion of lipid. Here the lipids are dispersed into smaller droplets where the surface tension is reduced and the surface area is increased. So the process is divided into three phases, that's detergent action of the bile salts, the surfactant action of the degraded lipids, and the mechanical mixing which occurs due to peristalsis. Coming to the detergent action of the bile salts, here the interaction with the lipid particles and the aqueous duodenal contents occurs, which convert the large lipid masses into smaller emulsified droplets. And they also stabilize the droplets by preventing their coalescence together or sticking of them together. The surfactant action of the degraded lipids. Here the products of the lipase action of the lingual lipases, like the free fatty acids and the monoacylglycerols, they act together with the phospholipids to behave as surfactants. They will reduce the interfacial surface tension and they will increase the water-lipid interface. Then we have the mechanical mixing of peristalsis. It's nothing but the physical churning which breaks down lipid into smaller droplets. And it produces better substrates for enzymatic digestion of lipids. Coming to the second part, that is digestion of cholesterol esters and phospholipids. Here, the pancreatic secretions, they also contain two more enzymes, that is one, the cholesterol esterases, and two, that is phospholipase A2. The cholesterol esterases will hydrolyze the cholesterol ester into cholesterol plus fatty acid, and the phospholipase A2 will hydrolyze the phospholipids into lysophospholipids plus fatty acids. The products of lipid digestion here are short chain fatty acids, medium chain and long chain fatty acids, 2 monoacylglycerol, 1 monoacylglycerol, glycerol, cholesterol and lysophospholipid. Coming to the second part, that is absorption of lipids. Here this will again occur in the two phases, that is direct and indirect. The direct absorption of glycerol, small and medium chain fatty acid occurs directly from the intestinal lumen, which enters the portal vein and it is transported to the liver, immediately utilized for energy. The second is the indirect absorption or absorption via the bile salts. The substrates absorbed are 2 monoacylglycerols, long chain fatty acids, small amount of 1 monoacylglycerol, the cholesterol, phospholipids and lysophospholipids. Here all these substrates are incorporated into the bile salt micelles, which are then absorbed into the intestinal epithelial cells. Now you can write key points, clinical aspects. Abnormalities in the absorption. One is styria, that's the fatty stool. It's characterized by foul-smelling fatty stool due to excessive excretion of the fat or lipids. This is due to the defective digestion or absorption, or it can be because of both. Then we have the defective absorption, which can be seen in the celiac disease, Pruia-Conn's disease, when the surgical removal of intestine is seen, and also when there is obstruction of the bile duct. Then we have chyluria. It's an abnormal connection between the urinary tract and the lymphatic drainage, which makes the urine appear milky due to the lipid droplets. So this covers your another 5 marker on digestion and absorption of lipids. Let's move on to the next question. That is classification of proteins based on function with suitable examples. Now the word protein is a Greek word which comes from proteos, which means primary. Okay. So proteins are polymers which are made up of amino acids which are linked by the peptide bonds. So how does the formation of the peptide bond occur? When an amino group of one amino acid combines with the carboxyl group of another amino acid, it forms a peptide bond. Now based on the number of amino acids we have, if it's 2, it's called as di. If it's 3, it's called as tri. 4 is tetra. Few amino acids are called as oligo. Now if we have about 10 to 50 amino acids, they are called as a polypeptide. Now when the long chains of polypeptides which are greater than 50 amino acids is called as a protein. Okay. We have the structural classification of protein also. It's rather complex, which can be divided into 4 levels. Primary, it's a linear sequence of amino acids. Secondary, the spatial arrangement is due to twisting and folding of the polypeptide chains. The tertiary is a 3-dimensional structure of a single polypeptide which forms a functional protein, whereas the quaternary structure is a spatial arrangement of 2 or more polypeptide subunits. Coming to the functional classification of proteins, we have the catalytic proteins which catalyze the biochemical reactions, like we have the hexokinase and amylase. We have the defense proteins which provide us immunity, like the immunoglobulins. Then we have the structural proteins which provide support and strength, like the keratin, hair, nail, and the collagen. Then we have the hormonal proteins which regulate the physiological functions, like the growth hormone, insulin. Then we have the contractile proteins which cause the muscle contraction, like actin, myosin, tropomyosin. Then we have the transport proteins which transport substances in the blood, like we have serum albumin and transferrin. Then we have the storage proteins which store essential substances like ovalbumin and gluten. Then we have the visual proteins which are responsible for vision, like we have the rhodopsin and iodopsin. We have the membrane proteins which transport ions across the membrane, like we have the sodium-potassium ATPase or the sodium-potassium pump. We have the hemostatic proteins which cause blood clotting, like the fibrinogen and prothrombin. We have the buffer proteins which maintain pH, like the plasma proteins and hemoglobin. The respiratory proteins which transport oxygen and store them. We have the hemoglobin and myoglobin. We have the receptor proteins which receive and transmit signals, like we have the insulin receptor, glucagon receptor, steroid hormone receptors. Then we also have genetic proteins which regulate the expression of the genes, like histones and transcription translation factors. So these are about 40. You can make a flow chart or also explain them in detail. So these are the few proteins. So this was the functional classification of proteins with example for 5 marks. We'll see the next 5 marker, that is glycogenesis. We will split this word into 2, that is glyco is glycogen. Genesis means synthesis. So it means synthesis of glycogen from the glucose molecule is called as glycogenesis. Glycogen is the storage form of glucose in the body. The site for glycogenesis is the tissue. We have the liver and muscles, and intracellular we have the cytosol of the cells. The starting compound is glucose while the end product is glycogen. Now we have the reaction pathways. Now we have divided the process into 4 parts. One is the synthesis of UDP glucose. The enzymes hexokinase in the muscle and glucokinase in the liver will convert glucose to glucose 6-phosphate. Then we have the phosphoglucomutase, which will catalyze the conversion of glucose 6-phosphate to glucose 1-phosphate. Then we have UDP glucose pyrophosphorylase, which will convert the formation of UDP glucose from glucose 1-phosphate with the help of UTP. Okay, don't worry, we will see this all reactions with the help of a flow chart. The second one is the requirement of a primer. To initiate the process of glycogenesis, the primer would be a pre-existing molecule of glycogen, which will be called as glycogenin. Then we have the glycogen synthesis with the help of glycogen synthase. Now, this is an enzyme which is responsible for the formation of 1,4-glycosidic linkages from the UDP molecule at the non-reducing end. Then we have the formation of branches in the glycogen molecule with the help of glucosyl alpha 4,6 transferases. This will transfer the carbon bonds from 1,4 to 1,6. Okay, so we have 4 steps, which we will see with the help of the flowchart. The first one is the synthesis of UDP glucose. Now here we have the glucose molecule. Now this glucose is converted to glucose 6-phosphate with the help of glucokinase. Now this glucose 6-phosphate by the enzyme phosphoglucomutase, which causes the transfer of phosphate group from 6 to first position, will form glucose 1-phosphate. This glucose 1-phosphate with UTP in the presence of UDP glucose pyrophosphorylase will form UDP glucose. This UDP glucose is nothing but uridine diphosphate glucose. This is very important for the process of synthesis of glycogen because it is a activated sugar molecule or activated glucose molecule, also called as the universal glucose donor. Okay, now this UDP, to form glycogen, we need a primer. So this is the second step. That is the formation of a primer, that is a glycogenin. Now, a pre-existing glycogen molecule is called as a glycogenin. It acts as a primer. The OH group of this glycogenin, okay, will act as a site for attachment of this initial UDP glucose molecule. This will occur with the help of an enzyme called as glycogen initiator synthase, which will transfer the first molecule of glucose, that is UDP, to the glycogen primer. Now coming to the third part, that is the formation of 1,4 alpha bonds. Now this glycogen primer with the help of glycogen synthase will form alpha 1,4 bonds at the glycogen primer molecule, and it will start elongation process. Okay. Then with the help of the enzyme glucosyl alpha 4,6 transferase, okay, we have this is the non-reducing end. Now 5 to 8 molecules from this non-reducing end will transfer and form the alpha 1,6 bonds and form a branched structure. Why do we have these 1,6 bonds is that, one, they increase the surface area, okay, for absorption. Two, the number of non-reducing ends are increased. Number of non-reducing ends are increased, okay, which will allow for rapid synthesis and breakdown of synthesis plus breakdown of glycogen for release of energy immediately. Okay. So now this process of 1,4 and 1,6 bonds continues till you form the glycogen molecule. The glycogen molecule is the storage form of glucose. So this covers your 5 marker on glycogenesis. Coming to the next question, specific dynamic action of foods, yeah, SDA. It's the phenomena of the extra heat that is produced by the body over and above the normal calculated caloric value when the given food is metabolized in the body. Suppose a body is producing about 130 kilocalories of energy to digest about 25 grams of carbohydrates, which would require about 100 kilocalories approximately. So the extra heat of 30 kilocalories that is produced is called as SDA. It's also known as the calorogenic action or thermogenic action or the thermic effect of food. The SDA values for protein is 30, lipids is 15 and carbohydrates is 5%. That's the structure of tRNA. tRNA is also called as the transfer RNA or soluble RNA. It has a clover leaf structure. It has 4 arms. First is the acceptor arm at the 3' end. The end sequence carries amino acids with the end sequence of CCA at the 3' end. The second arm is the anticodon arm, which is the opposite of the acceptor arm. It has the base pairs complementary to that of the mRNA. The third arm is the DHU arm or the D arm, or also called as the dihydrouridine arm. It serves as a recognition site for the enzymes. Then the fourth arm that we have is the pseudouridine arm, which is involved in binding to the ribosomes. We'll see this with the help of a diagram. This is your 5' end and this is your 3' end. As you see, the end sequence is CCA. Okay, this is the amino acid end. Opposite to the acceptor arm, we have the anticodon arm. Okay, which is the binding site for mRNA. Then we have the dihydrouridine arm or the DHU arm, which is the binding site of enzymes. Then we have the pseudouridine arm, which is involved in binding to the ribosomes. The next question is competitive inhibition of enzymes with examples. The inhibitor, which is closely resembling to that of the real substrate, is regarded as a substrate analog. The inhibitor competes with the substrate and it will bind with the active site of the enzyme, but it does not undergo catalysis. As long as the competitive inhibitor will hold this active site of the enzyme will not be available for the substrate to bind. Here the Km value increases and the Vmax remains unchanged. So we will see this with the help of a diagram. This is your enzyme plus now we have a substrate, okay, and we also have an inhibitor. This is your substrate that was actually supposed to go and bind here, but instead of the substrate going and binding, we have this inhibitor which is going and binding to the enzyme. Okay, now this inhibitor has gone and bound to the active site of the enzyme, making the substrate unavailable for binding. This is called as competitive inhibition. We will see examples. We have the succinate dehydrogenase, which is the enzyme, which is going to bind with succinic acid, which is the substrate. Okay. But we also have inhibitors like malononic acid, the glutaric acid and oxalic acid, which have the same structural similarity, which will bind at the active site of the succinate dehydrogenase enzyme. Then we have the enzyme alcohol dehydrogenase. Okay. Methanol poisoning time पे when this enzyme has to go and bind. So we have ethanol, which will act as an inhibitor to the substrate, that is methanol. for the enzyme alcohol dehydrogenase. So it's used in the treatment of methanol poisoning. Other examples we have the xanthine oxidase enzyme. The substrate was supposed to be hypoxanthine and the inhibitor is allopurinol. Then we have the monoamine oxidase. We have the catecholamines, epinephrine and amphetamine will be your inhibitors. Then you can see the vitamin K epoxide reductase is the enzyme. The substrate had to be vitamin K, whereas the inhibitor is dicumarol. Then we have the HMG CoA reductase, where we have HMG as the substrate, lovastatin and pravastatin as the inhibitors. The next two markers, that is the blood buffers and their significance. A blood buffer is the one which maintains the pH of blood between 7.35 to 7.45, which prevents any dangerous shifts by neutralizing any excess or acid during the metabolism process. So the blood contains three buffer systems. That's the bicarbonate buffer, which has the sodium bicarbonate and carbonic acid. It maintains the blood pH of about 7.4 and the ratio of bicarbonate to carbonic acid is about 20:1. Then we have the phosphate buffer. Here we have the sodium dihydrogen phosphate and disodium hydrogen phosphate. They act intracellularly and maintain the phosphate levels of about 6.8, which is more important in renal tubules and intracellular fluid. Then we have the protein buffers or the plasma proteins like hemoglobin. They together constitute the plasma protein buffer system by maintaining the H plus ions. Then we have the plasma buffers. They're very minimal or almost null, but they act through the proteins of albumin, which can accept or donate again the H plus ions. They usually act in combination with the protein buffers. The significance is that they maintain the acid-base balance, they prevent sudden changes in pH, they support enzyme function and metabolism, they facilitate oxygen and carbon dioxide transport, and protect against acidosis or alkylosis. Come to the last two marker, that is the coenzyme form of thymine. We have thymine pyrophosphate. Riboflavin, we have the flavin mononucleotide. Niacin, we have NAD and NADP+, that's nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. Then we have folic acid. The coenzyme form is the tetrahydrofolate. So with this, we come to the end of the 2025 biochemistry paper. Stay tuned for our October 2025 physiology paper discussion. Like, share, and subscribe for more such videos. You can also join our Telegram channel for previous year question papers, solve dental PYQs, and also for full video description links.